Hello everyone! This section is for bacterial plasma membranes. After learning this section, you should be able to describe the fluid mosaic model of membrane structure and identify the types of lipids typically found in bacterial membranes. The cell envelope is defined as the plasma membrane and all the surrounding layers external to it. The cell envelopes of many bacteria consist of the plasma membrane, cell wall, and at least one additional layer, such as capsule or slim layer. Of all these layers, the plasma membrane is the most important because it encompasses the cytoplasm and defines the cell. If it is removed, the cell's contents spilled into the environment and the cell no longer exists. Furthermore, despite being the innermost layer of the cell envelope, the plasma membrane is responsible for much of the cell's relationship with the outside world. Thus, we begin our consideration of bacterial cell structure by describing the plasma membrane. First, let's talk about the function of plasma membranes. First, a primary rule of all plasma membranes is that they are selectively permeable barriers. They allow particular ions and molecules to pass either into or out of the cell, while preventing the movement of others. Thus, the plasma membrane prevents the loss of essential components through linkage, while allowing the movement of other molecules. Second, bacterial plasma membranes play other critical roles. They are the location of several crucial metabolic processes, such as respiration, photosynthesis, and synthesis of lipids and cell wall constituents. In addition to plasma membrane, some bacteria have extensive intracytoplasmic membrane systems. These internal membranes and the plasma membrane share basic design. However, they can differ significantly in terms of the lipids and proteins they contain. To understand these chemical differences and the many functions of the plasma membrane and other membranes, it is necessary to become familiar with membrane structure. Okay, let's look at the structure of plasma membrane. The most widely accepted model for membrane structure is the fluid mosaic model, which is proposed that membranes are lipid bilayers, within which proteins float. You can see them on the picture. The model is based on studies of eukaryotic and bacterial membranes and was established using a variety of experimental approaches, including transmission electron microscopy and atomic force microscopy. Cell membranes are very thin structures, about 5 to 10 nanometers thick, that look like two dark lines on either side of a light interior when imaged by TEM. You can see it on this picture. The characteristic appearance is evidence that the membrane is composed of two sheets of molecules arranged end to end. Cleavage of membranes by freezing etching. It is a technique that allows the microscopist to see fine detail. The result exposes the protein lying within the membrane lipid bilayer. The chemical nature of membrane lipids is critical to their ability to form bilayers. Most membrane-associated lipids, such as phospholipids shown in this picture, are amphipathic. They are structurally asymmetric, with polar and nonpolar ends. The polar ends interact with water and are hydrophilic. The nonpolar hydrophobic ends are insoluble in water and tend to associate with one another. In aqueous environment, amphipathic lipids can interact to form a bilayer. The outer surfaces of the bilayer are hydrophilic, whereas hydrophobic ends are buried in the interior, away from the surrounding water. Two types of membrane proteins have been identified based on their ability to be separated from the membrane. Peripheral membrane proteins are loosely connected to the membrane and can be easily removed. They are soluble in aqueous solutions and make up about 20 to 30 of total membrane protein. The remaining proteins are integral membrane proteins. You can see them on the picture. These are not easily extracted from membranes and are insoluble in aqueous solution when freed of lipids. Integral membrane proteins, like membrane lipids, are amphipathic. Their hydrophobic regions are buried in the lipid, where the hydrophilic portions project from the membrane surface. Integral membrane proteins carry out some of the most important functions of the membrane. Many are transport proteins used to move material either into or out of the cell. Others are involved in energy-conserving processes, such as the protein found in electron transport chains. Those integral membrane proteins with regions exposed to the outside of the cell enable the cell to interact with its environment. Then let's see the bacterial lipids. Bacterial membranes are similar to eukaryotic membranes in that they are lipid bilayers, and many of them, amphipathic lipids, are phospholipids. The plasma membrane is very dynamic. The lipid composition varies with environmental temperature in such a way that the membrane remains fluid during growth. For example, bacteria growing at low temperatures have more unsaturated fatty acids in their membrane phospholipids. That is, there are one or more double covalent bonds in the long hydrocarbon chain. At higher temperatures, their phospholipids have more saturated fatty acids, those in which the carbon atoms are connected only with single covalent bonds. Okay, that's all for this section. Here is the word list for you.